You can understand how DNA mutations manifest into a final protein product by imagining that our cells are tiny restaurants. DNA is a customer who wants protein to be made, so he makes an order by telling the cashier the coding strand sequence. The cashier needs the POS system to convert the coding strand sequence to mRNA by replacing the T's with U's. The mRNA is a line of tickets that Chef tRNA reads to help translate them into amino acid meals that make up the customer's protein order. So DNA tells the cashier the sequence of the coding strand from 5' to 3' direction. The cashier enters the exact sequence that DNA asked for into the POS system and sends it to the kitchen. Once it reaches the kitchen with the T's changed to U's, it is now mRNA that is ready to be translated by Chef tRNA. Each codon is printed on its own ticket. He has to recognize the start codon, which resembles the start of a new order, and the stop codon, which must be present for the chef to know when the DNA's protein order is complete. All of the amino acid meals make up the protein that the DNA ordered. In the case of a point mutation, the DNA accidentally gives the cashier a coding strand sequence with an incorrect nucleotide. A missense mutation is when the incorrect DNA nucleotide results in one of the amino acid meals being replaced by a different one, making incorrect protein orders. In this case, the G to C mutation is a transversion mutation that results in the chef making serine instead of arginine. Nonsense mutations are when the incorrect nucleotide makes a stop codon from what was originally coding for an amino acid. This example is an A to T transversion mutation. The chef reads the DNA sequence entered and only makes amino acid meals up until the UAG stop codon, which was previously AAG, coding for lysine, but now this amino acid meal is missing in the protein order. Silent mutations are when the nucleotide error results in a different codon but still codes for the same amino acid meal as the original codon. GTC to GTA is a transversion mutation. Because both GUC and GUA code for valine amino acid, the chef makes the correct protein despite the mutation. Read-through mutations occur when the incorrect nucleotide turns the original stop codon into one that codes for an amino acid. TAA to AAA is another transversion mutation where the stop codon becomes a codon that codes for a lysine. The removal of the stop codon means the following codons will be made by the chef as a part of the customer's order, resulting in extra amino acid meals in the order.